Here we go. So welcome everyone, and welcome to our monthly virtual series. My name is Sandra Franco, and I am the Education and Outreach Coordinator for the Center for Integrated Cellular Analysis. And for those of you that are not familiar with the center, we are a collaboration, or we represent a collaboration between 9 labs across New York City. And we are interested in developing methods and tools focused on multimodal single-cell analysis. This seminar is part of our recently created virtual series, where we aim to highlight methods, research papers, and preprints and manuscripts that stem from our center, and that we think that are interesting for the broader biological community. Before starting, I just want to go over a couple of organizational details. So today we will have one talk, and at the end of the talk, there's going to be some time for you to ask questions. You can either raise your hand and I'll call on you, and you can unmute yourself and ask the question. Or if you prefer that I ask the question to our speaker, you can also type the question in the chat box, and I'm going to read it to Aidan. With that, I'd like to introduce Aidan Daly today. Aidan is a research fellow jointly appointed between the Center for Computational Biology at the Flatiron Institute and the Center for Genomics of Neurodegenerative Diseases at the York Genome Center. His work focuses on the use of spatial transcriptomic and proteomic data in concert with computer vision and Bayesian modeling to understand the spatiotemporal dynamics of disease, ALS. Aidan, I'll pass it over to you.
All right, great. Yeah, thanks for having me, everyone. I'll go ahead and start sharing my slides here. And yeah, I'll just launch right in. So today my talk is going to be common coordinate registration of higher resolution histology images. So I'll first kind of explain why we should care about this and what common coordinate registration means in this context, and why I think it's an important thing, particularly in the field of spatial-omics. So starting right off, so when we're studying tissue, obviously spatial context is very important. So we see on an example image on the lower left where we have neural tissue stained for a couple different markers of different cell types. Tissue is a very complex patterning of different cells of different types that are all interacting with each other. So when we're measuring any sort of quantity of interest within a cell, mRNA or protein, we will have very different interpretations depending on the cell type from which it originated and the location and context of that cell. So, while we were starting off as the field of RNA analysis, for example, we started with bulk RNA, where we would, take this nice tissue, throw it in a blender, and we would get all the RNAs that we were interested in, but we lose both the cellular and spatial context. Nowadays, we have this nice single cell technology where we can use sort of microfluidics or bead devices to separate out individual cell transcriptomes, which is nice. We get a lot more context about where each RNA profile can be assigned, but we still lose the native spatial context. So moving forward, a sort of newer approach to this is spatial transcriptomics, or proteomics. Works on a very similar methodology, so you could just call them omics technologies. And there's basically 2 flavors of this at the moment. So on the left here, we have our FISH-based or fluorescence in situ hybridization-based approach, which is based on using targeted fluorescently labeled probes in situ to label subcellular targets, any mRNA species or protein species of interest, and then to image them via microscopy. So it's very nice because it has a very good, subcellular resolution. You can look at organelle level placement of mRNAs or proteins. It has a very high signal to noise ratio. Even very rare species can be reliably detected on modern hardware. The problem with this, though, is that you have to know your targets in advance, and the sort of plexity, the number of targets you can look at any one time, is limited to sort of several dozen, for reasons of overlapping image spectra or emission spectra that limit how many fluorophores you can use at a single time, you have to keep washing and re-imaging, and tissue will degrade over very long image acquisition periods. So on the right, we have a technology that has a different approach that addresses some of these issues and has some issues of its own. So this spatially barcoded approach to spatial transcriptomics operates on, again, intact tissues. So in this case, I'll talk through the example of the ST or Visium workflow, but all of them are somewhat similar. You'll take a thin section of tissue, about 10 microns thick, and you lay it onto a slide that has been specially printed with this array of spatially barcoded spots. And each of these spots contains a field of poly-T capture probes that are fused to the slide rather. When the tissue is placed and permeabilized, mRNA will fall down and hybridize onto these probes. You can then cleave these probes, reverse transcribe into cDNA, do your library amplification sequencing. And because of these little spatial barcode sequences, you can map every read back to a coordinate in space. So this is it's an unbiased measurement of the transcriptome. It captures all mRNAs nominally with the same efficiency. So you don't have to specify your targets in advance, and you get thousands of unique genes at once, depending on the technology. The downside of this is the resolution is a lot coarser than the fish-based methods. It's basically, as coarse as we can print these spots together. So this can range anywhere from 10 to 100 microns, depending on the technology you're using. And it's also susceptible to some technical error. There's a chance of dropout events, which are basically mRNAs that are present in the tissue, but for whatever reason are not captured by the probe. So these are the pros and cons of each technique. So when we were looking at these, it's a very nice picture, oh, great, look at the distribution of RNA in space. But if we actually want to reason about sort of trends at a level of an individual or even a population, certainly, we're going to need to integrate data from multiple sources, even multiple replicates of a single individual could help us overcome some of the technical noise that's associated with ST. So the problem with this is that biological and technical variation from sample to sample really complicate the idea of sort of mapping one region of tissue onto another. So like a spot in an ST experiment is really dependent, on if there's local damage to the tissue, how the tissue is loaded onto the array, if the same exact part of tissue overlaps a spot or not. There's really hard to make a one-to-one mapping. So this is where registration comes in. So registration in this context is a transformation that maps coordinates in one system, say one tissue, onto coordinates in corresponding coordinates in another system, another tissue. So if we're looking at serial imaging, where we're looking at the same tissue imaged in multiple rounds, as we might with fish, then if we're trying to make one cohesive picture of all these serial images together, we can use subcellular landmarks like DAPI stained nuclei as like key points to anchor ourselves between rounds. But if we're performing across samples, even if we have these DAPI nuclei, if we're trying to register one nucleus onto another, and there's no real sort of one-to-one correspondence, we don't have these cellular markers to rely on. So what do we do instead? Well, that's where common coordinate registration can help us. So in the absence of a conserved object, we look instead for conservation of higher level features. So if we were to take a tissue, here we have a cross section of a mouse spinal cord, and we partition it into different anatomical regions. We have gray and, basically sort of gray matter in the center and white matter around, but the positioning of the gray and white matter on the, bilateral and dorsal ventral axes change the tissue type that we expect there. So if we were to take some example mouse spinal cords, label them at regions of interest, in this case, these are all spots in an ST experiment, we can make a correspondence across tissues. So what does this enable us to do? So at a very basic level, this enables us to just integrate data from experimental replicates. So here we have a sort of scatter plot of a lot of different ST experiments done on a couple hundred different mouse spinal cords that have been aligned such that the centroids of their dorsal and ventral horn regions overlap. And we see that, from what was a very coarse, 100 micron center-centered, or actually 200 micron center-center distance, becomes a pretty good coverage of the tissue and gives a pretty clear idea of what the general structure is. if we're to look at what we get from the transcriptomics end of this, we can actually use this to correct for that sort of lack of signal that we see relative to fish-based methods. So here's an example where we have several, I'd say a couple hundred mouse spinal cords, ST, that have been registered with common coordinate registration and projected into this space. And we see that the distribution of these marker genes, which we captured in an unbiased way, recapitulates the distribution in a targeted immunofluorescence experiment. And we can use this also with a sort of larger, more interesting cohort to compare sort of how disease-related markers change expression patterns over space and time. So we have two marker genes here, AIF1 and GFAP. If we're looking at a wild type mouse versus a SOD1 mutant mouse, which is at two different time points, we see that the distribution of AIF1 expression becomes really dysregulated, particularly down here as the disease progresses, and the same with GFAP. But beyond just making pretty pictures, this actually enables us to do some pretty cool statistical analysis on a really big, diverse cohort of data. So I'll just quickly touch on this paper, Splotch, which you can see I put a citation enough down there in Science. So Splotch is a hierarchical Bayesian model of spatial gene expression that operates on registered data. So if you have a bunch of in this case, spinal cords, but whatever tissue you're interested in that's been registered to a common coordinate framework, what Splotch basically does is it aligns these data based on the AAR tags and uses them to make a sort of estimate of what the characteristic expression is of each gene in each region. It then sort of models the actual expression of each gene as a combination of the effects of this region-specific expression, as well as effects of local neighborhoods of other spots and spot-specific random effects. And the region-specific expression is formulated hierarchically so that you can sort of stratify different samples based on genotype, age, sex, et cetera. So you could start with a bunch of mice and have mouse-specific expression, then average over all your male mice of a certain genotype to see what's your male-specific expression. Then your genotype-specific expression time point. It's a very flexible, cool formula that depends on common coordinate registration. So enough motivation, let's talk about actually how we do this. Well, the way we actually did this to start was just by labeling every region of interest by hand. But as the number of tissues that we're looking at and the resolution at which we can probe the transcriptome increase, this is not going to hold up. So let's do some, nice machine learning approaches. So there's two basic approaches to doing this currently. So one is a semantic segmentation approach, which is basically, can we associate from an image, can we associate each image pixel or superpixel with a class label? This is on the surface is exactly what common coordinate registration is. On the right here, we have a diagram of UNet, which is a popular segmentation network where we take an input image. In this case, it's a bright field image of some cells. We put it through a couple convolutional layers, which are just kind of scan the image and extract some higher level features from it. We keep doing this all the way to get a very abstract representation. And then we combine this abstract representation with some feed forward connections that maintain low level information about the original image. And we produce our best guess at these different objects, location and spread and label. And then we train this network. If it's off, we have our error that we back propagate through the network, and we hope we do better on the next pass. So the problem with this is that maintaining these sort of subcellular resolution features requires that we maintain a lot of pretty big amount of state in memory at once while we're training the network. So for a very high resolution image that covers a lot of space, we will run out of memory very quickly while we're training. So the ways that you can fix this are either to reduce the complexity of the network, so make it less, reduce the number of parameters so there's not as much to keep in memory at once, but that reduces our ability to detect sophisticated patterns. The other option is we reduce the complexity of the input. We either downsample the image, decrease the resolution, or subsample it. We chop it into bits and process the bits independently. Either way, we lose our ability to kind of maintain a global view or a subcellular view. We're trading off to keep those happy. The other approach, patch classification. So this approach decides to go fully local. If segmentation tries to be as global as possible, patch classification is as local as possible. We extract one image patch at every region of interest. So it could be at the locations of our ST probes, and we run it through a CNN that eventually predicts the class assignment. So these are actually, as you might expect, pretty prone to local error. For example, if the region of the tissue here is damaged, then you might not be able to predict accurately what type of tissue this is, because you don't have any context to correct for that damage, that error. One thing you can do to help this is you can add useful covariate information. So if I was trying to classify the spot, and I also told the network where it is in some sort of common coordinate framework, that would help to correct for that error. But that's a very problem-specific thing. What if we don't care about the spinal cord and we want to look at brain? Or what if we want to look at a tumor which has no stereotyped anatomy? This won't work. So we want something instead that has a nice global context so we can correct for local error, that has enough, of a local view that we can pick up on subcellular features that are important, and that can generalize to almost any tissue that we're interested in. So this prompted us to formulate this hybrid approach that we've been calling GridNet. So the way GridNet works is takes an image, a histology image. We sample patches at our regions of interest. So in this case, it could be the locations of ST spots in an experiment. We run each patch independently through a patch classifier, some CNN that can take these subcellular features and extract useful information from them. And we make our predicted vectors, if we have 10 classes, this is the probability of being in each of the 10 classes based on that patch alone. We then stitch these vectors back into a 3D image, which shows a kind of subsample or sort of downsampled, very abstracted view of the tissue, where each sort of pixel in this case contains a vector saying how likely we are to assign that pixel to each of the classes. We then run it through a segmentation network, or corrector network, that scans over the image. It pulls information from neighboring pixels to update the belief in the assignment of a certain pixel of interest. And finally, we get an output map. So the idea is that we have the patch classifier that looks at subcellular scale and the corrector that looks at a global scale. So we tested this on two data sets. One was an Allen Brain Atlas reference histology data set, which is basically a couple hundred mouse brain cross sections where different regions have been assigned to different tissue classes. And I picked a certain ontology level that gave us 13 classes across the whole data set. And basically, this is a simulated ST data set that is very nicely put together. The staining is consistent, the sample orientation is consistent, we like it. The next one we have is an experimental ST data set from this mouse spinal cord assay that we've been talking about. So it's 400 mouse spinal cord arrays that have actually been processed with spatial transcriptomics and have been manually registered to a common coordinate system with 11 classes. This one shows a lot more variability in sample orientation and staining. So we'll get to see how this GridNet handles these effects in real life. So to compare this to see that we're actually improving on what we can do, we compared it to two sort of competing approaches. One is just a pure patch classification approach with the DenseNet, which is a very powerful, popular architecture. Basically, again, you take your input, it passes through many different convolutional layers, extracting more and more features, and you get your output. DenseNet is actually quite nice because it basically every layer adds to a kind of a shared state. And it actually ends up saving parameters because you don't have to sort of reprocess the input at every single stage. You just pass forward a shared state and every layer adds on a bit more information. So this F network basically applied to just all the foreground patches, bing, bang, boom. That's the patch classification approach. The next is a pure segmentation approach. So in this case, we took, we wanted to take something that could actually fit into memory and look at the entire global view of the tissue. In which case things like UNet, which are very apparently have a lot of parameters, just weren't going to be able to cut it. Even a ResNet 34, which we adopted to do segmentation. So normally ResNet works kind of similar to DenseNet. Take your image, you go through all these different connections, and in a normal case, eventually it flattens it and predicts the class. But in our case, we use this sort of abstracted feature map to predict our registration. So we just kind of map from this feature space onto our desired ST dimensions. So it takes a whole image as an input, and the segmentation mask is our class assignments that are used to train it. And to even fit just one image from the Allen Brain Atlas into this network, we needed to drastically reduce the complexity of every layer. So this kind of just highlights the difficulty of maintaining a global view with even a decently simple segmentation approach. So here's some of the results that we got from this comparison. So we have from both data sets, the Allen Brain on the top, Maniatis here. We show the results from our patch classifier, DenseNet, our segmentation approach, or ResNet, and the GridNet, which does a little bit of both. So what we did for this was we trained each network 10 times on the training data, and we kept the 5 best that performed on a validation set. And then we reported the mean, standard deviation, and ensemble performance of those 5 best on a held out test set. So we can see that the GridNet does vastly better than the ResNet. And the ResNet actually does a lot worse even than just the patch classification alone. And we are pretty sure that's because of how much we had to simplify the network to just fit the problem into memory. So, but even compared to the sort of patch classification approach, we get a noticeable improvement by adding this second layer. We get a much better accuracy and AUPR across every class in the data set. Same thing with the Maniatis data set. We see the same pattern with a slightly lower kind of best performance that is due to these sort of experimental variations. We have varied staining to kind of confuse nets. They think even if you're looking at the same tissue that is stained differently, it has to figure out that it's the same, and sometimes it can't. So we were very happy about this performance. We did notice that a couple classes had a little bit noticeably lower AUPR. So in the Allen Brain Atlas, the cortical subplate, in the Maniatis data set, things like the lateral central canal, that was a doozy, lateral edge. And we found, sort of going on to the next slide, that the problems that GridNet still has might be hard or kind of impossible to solve. We're reasonably confident that GridNet's doing about as good as we can on these tissues. And the way that we determined this was looking at the predictions at tissue boundaries, so regions where 2 different types of tissue meet. So we found that applying GridNet greatly reduces the chances of making an error in a region of contiguous tissue. So here I have these sort of prediction accuracy maps, which is basically in each one, we show the tissue in this kind of like Space Invaders thing, where every pixel is a spot in an ST experiment, and regions between tissue types are colored in as white outlines, and every pixel shows the probability of misclassifying the patch. So basically the sum over all the incorrect classes that is spit out by the model. So in the Allen Brain Atlas, if we just use the F network, just use our DenseNet, we make these point errors where, because maybe, just noise in the data or damage in the tissue, it makes a mistake, or it's likely to make a mistake. After we apply the second layer of the network, all those errors go away, and the only errors we make are at regions between tissue types. And the same thing is in the Maniatis data set. It's a little harder to see because the tissues are smaller, and there are sort of fewer regions of huge contiguous tissue, but it's the same pattern you see. So 2 explanations for this. One, label noise. Like maybe it's very hard to reliably assign tissues to one or the other because they have an intermediate identity at these boundary regions, in which case we can't really do much about that. That's a failure of the common coordinate system. The other case is that also in any uncertain situation of uncertainty, which is inevitable, the model might predict the more likely tissue class. So if it sees like tissue A and tissue B, and tissue A is 4 times as likely as tissue B, if it's a toss-up, why not pick tissue A, get the best chance? So these types of things can actually, even if you have no noise and all the data are annotated as best they can, you might just have some error against the rare tissue classes. And that's something you can correct for by adding weights to your loss function that says, no, say this is a rare cancer subtype and we really want to get it right. You can tell the model that in advance. The experiments we did not do that, but it's a fun thing to know. So briefly, I'm going to talk about sort of how this approach can scale with increasing resolution. So the Visium platform is the new and popular way to do ST, and it's a lot more, it has a lot more resolution, or a lot greater resolution than the data we've been looking at so far. The size of the spots that we're looking at has decreased from 100 microns to 55 microns, and it's packed in a hexagonal grid. It's like a honeycomb, which is a greater packing efficiency than 4 squares. So a lot more spots per tissue area. So the problem with this is that convolutional neural networks are designed for data that are packed into nice regular Cartesian grids. You take a matrix and you scan it over your data row by row. In a hexagonal system, that doesn't quite work. If you were to basically represent this honeycomb as a regular grid by just shifting up every other column, then taking a matrix and scanning it over doesn't actually show the true 6 neighborhood of the spot. This spot 6 here should have neighbors 2, 5, 10, 11, but not 1 and 9. So what you need is this weird kernel in that doesn't look like a regular matrix to actually get the true neighborhood of the spot. And it turns out you can do this by basically just having 3 different padded versions of the input, doing convolutions on them and merging them together. So it's basically, there's a hack that you can do to get a true hexagonal neighborhood representation in this, and we can easily update any network that we have to do hexagonal convolutions instead of Cartesian. The more interesting and really tough problem to manage was that even with this sparse sampling, taking several thousand image patches from this high resolution image is still a lot of data, and it will quickly overwhelm the GPU if you try and train this network end to end. So, this may not have been a case when you're sampling very sparsely, but as we go to Visium arrays where you might have 5,000, up to 5,000 spots per image, this will become a problem. Future technologies might get even denser. So what we do is this thing called gradient checkpointing. So to do that, I'm just going to quickly at a high level explain how we train neural nets down here. So I have a little GIF that once it loops, we start with an input. As the input goes through our model, it's put into a bunch of different intermediate states, and we have a prediction, which we compare against our observation. And then at every intermediate layer, we backpropagate the error. So we update our parameters in response to how good or bad we did. Normally, you keep all these intermediate states in memory so that you can easily do this gradient calculation and backpropagation as you need to. But with with the size of the input being very big, that might not be possible. So what we do instead is we institute a checkpoint where we say, I'm only going to remember some fraction of these intermediate states that I've chosen in advance, and everything else I'll recompute on the fly. So in here, we say we go through our network, but we only remember this guy. And then as we are doing our backpropagation, we recompute these intermediate states as we need to. So we basically trade off time for memory. So this time memory trade-off allows us to fit a lot larger inputs in by basically just breaking up our input into chunks, but kind of the way that RAM does. We're not actually sacrificing any complexity. We're just being a bit more inefficient so that we can actually keep a whole view of the image. And then the other thing you can do is gradient accumulation. So if you can only fit one image into training at a time, you might have a very noisy training path. So what you can do instead is, instead of doing backpropagation at every image, you sum the gradients over several images. So you simulate a larger batch of data that you can actually fit into memory. So a quick summary of that. So basically, we proposed this hybrid classification segmentation approach where we have a classifier network that extracts features at, from full resolution image patches. And then we have a corrector network, a segmentation network that takes these features and pulls in neighborhood information to refine our prediction of what the tissue registration is. The remaining error after this concentrates heavily at tissue boundaries, which makes us think that we're doing a good job because we know that those are hard to predict. Again, if you have a reason to believe that there are certain classes that need to be predicted more likely than others, you can upweight those to say, like, make sure you get this one right. And this way you don't lose at the boundaries as much. And finally, this approach, because of these sort of memory hacks we've made, it can scale with increasing resolution. We've written an interface where you can use it directly on the Visium platform. And we have a preprint out. It's forthcoming in bioinformatics, which I'm happy about, and all the code is available on GitHub. So I'll just briefly talk about a couple future directions for this, and then I'll open the floor to questions. So the first thing is I've been talking mostly about ST data, and I've kind of thrown this fish data by the wayside, right? What if we want to apply this to fish? Well, for fish images, since we're just doing in situ staining, we don't have this regular grid that makes sense, right? The distribution of the regions of interest are, the regions of interest to us are probably cells, and cells can be distributed seemingly at random, certainly not in a regular grid. So how do we handle this? What we can do instead is we can formulate G as a graph convolutional neural network, where we basically say our data, our regions of interest I've highlighted are maybe these cells where we have a certain number of, read counts of a species of interest. And we formulate a graph convolutional network. Basically, instead of convolving over a regular grid, it starts by pooling information as a graph would. If this guy has these four as neighbors, because we've decided that there's neighbors are within a certain radius, we update our estimate at this point based on the estimates of these neighbors. And then we scan over the whole network doing the same thing. So it's still possible. It's less efficient because you don't get to do all the fancy matrix operations, but you can still apply it this way. The other thing is we've only been using, even though we have both histology and sort of transcriptomics data available, we've only been using the histology data for registration, which might be sufficient for some systems, but in some systems where there might be patterns that are invisible to the eye, you might want to leverage both. So there's a lot of networks that are being proposed now that can look at both simultaneously. This is an example of an unsupervised network from Aviv Regev's lab that she's been calling in situ vec. I couldn't find a preprint on this, but she mentions it in this cool talk here at the bottom, where basically you take, in this case, an image of your different spots, as well as the expression data that you have at those spots. And you kind of put them through this autoencoder, they talk to each other, and then you can get a sort of cleaned expression estimate by putting in this image data as well. So you could imagine that if we wanted to do a joint feature extractor, we could have these modalities talk in an interesting way. And finally, what do we do if it's really just fundamentally hard to label the tissue? What if we can't supervise this, but we still want to register tissue? So you can have situations like this where manual labeling is complicated by either inconsistent sectioning, where you might just be looking at things from different slice angles, and it's very hard to reason about where you are in space. Tissue that has like a lack of visual hallmarks, or tissues that exceed the bounds of the ST array, so you don't have this full context to help you put yourself in a certain location. These are all kind of true in this human dorsolateral prefrontal cortex sample that we're looking at. So here I've shown a view from the Loop browser, which comes with the 10x Visium platform, and they've sort of just taken all the expression data at every spot and just done like a k-means clustering in a reduced dimensionality space and mapped it back onto the tissue. And we see that, by and large, we do get these nice cortical layers, but it's very noisy because they're not using any image data and they're not using any spatial data. So if we could find basically a way to do these types of unsupervised or self-supervised approaches to labeling, but with incorporating this nice, neighborhood information and information from both modalities, that would be ideal. So all that's left again is to thank my advisors. So at the top, Rich at Flatiron and Hamali at the Genome Center, Christoph Garros at NYU. He's the machine learning guru who helped this whole thing happen. Silas is our go-to guy at the Genome Center, and then students Timur and Joanna who've done a lot of work on this stuff with me. So thank you, and I'll open the field up for questions now.
Thank you, Aidan. That was an amazing talk. So now, yeah, as he said, we are going to move to the Q&A section, and please, you can either raise your hand or you can type the question in the chat box. We have a couple of questions here. I think you already touched on Noah Greenwald question, but maybe you can comment a little bit further. So the question is, you show some hematoxylin and eosin images for the patch classification. Is the network taking in the SCT data and also the hematoxylin and eosin?
So yeah, no, this so far is just using H&E data, and that's why we were so happy that we got up to almost 90% accuracy on held out images with just H&E data. I am definitely thinking that in the future, particularly in cases where you don't have these clear visual hallmarks like you would have made in the brain or the spinal cord, say you're looking at a tumor biopsy, the H&E image alone might not be enough to give a reliable segmentation. So you might want to definitely look at both. So yeah, again, instead of basically you would just reformulate your patch classifier F to, instead of only taking in an image, take in paired image and ST data. Maybe you would look only at a few, like a couple hundred, like tens to hundreds of marker genes to help kind of bias exploration in the correct direction. But yeah, I think that's something that could definitely enhance registration accuracy if done correctly.
I think Joy Otten is raising a hand, so you can unmute yourself.
Okay, thank you. I have a question actually additional to this. I was wondering if you were able to use GridNet on Visium slides wherein the boundaries aren't clear, so they basically go over the grids. You
mean where it goes beyond the bounds of the Visium array?
Yes.
Yes. So we did, yes, we used this on the, Maynard had put out a data set of about 12 human DLPFC tissues, all of which exceed the bounds of the array. So yes, and it is able to be applied in those situations. The idea for those is that in the cortex, you'll see like a kind of classical layering of, here you go. It's like a sort of layering of cells. And you can see here sort of the tissue continues on beyond this. Every cortex looks kind of differently, but the sort of ordering of layers is always conserved. Like sort of layer 1 will only ever abut itself in layer 2. Layer 2 will only ever abut layer 1, layer 3, etc. And these are rules that GridNet can learn even if there's only partial data available. Basically, these sort of the neurons of what you would hope happens, and if there is a good signal, what will happen is basically each neuron in the corrector network will look for local learning rules. Like if I am a patch, that is surrounded by layer 1 patches, then it drastically increases my chance of being layer 1. And that could be true even if you don't have all layers present in the image, for example.
Okay, thank you.
We have here Denise Duma is asking for a clarification. So how do you combine the patch classifications from the first stage into the pixel features in the second stage? Yeah,
so basically we're doing it's kind of just like a dimensionality reduction technique in a sense, right? So we started with this image, which is, number of pixels wide by number of pixels tall. And we've now sort of sampled this grid that is, say, what I've been saying, like it's HST pixels tall and WST, pixels wide. In this case, our pixels are actually now whole images. So we're, super pixels in a sense. So this whole input is now like a HST by WST array of image patches, which are each, 128 by 128 by 3 pixels. And then each of those gets passed through this classifier, which spits out just a one-dimensional vector. So we basically transformed this 5-dimensional input into a 3-dimensional input, which just represents the, where the coordinate is now the coordinate in the ST array. and the information at each coordinate is a vector describing the likelihood of being associated with each class. Does that make sense?
I think it does to me.
Okay.
I have a question myself. So you have used GridNet in brain images. Is this method also applicable to tissues that maybe are less organized than the brain?
So yes, we certainly hope so. So we don't have any so part of the reason I made this approach was like I was considering, when we looked at a competing approach, which basically this patch classification approach kind of has the same shtick as GridNet, which is like we take patches from regions of interest and we classify them and we combine this with some sort of spatial information. But then again, like this was a cheat because this is a tissue where we have this conserved anatomy, but I wanted this to be applicable to things where there is no conserved anatomy necessarily, like tumors. So we didn't actually get to do any tumor data, but we did find, like I said, that these cortical data were actually quite helpful because most of the tissues only had, a lot of the tissues only had some subset of the layers and were presented in different orientations. So one would hope that if we were in a completely unstructured tissue that had, distinct tissue types within it, you'd still be able to learn these patterns based on just a combination of a bunch of simple rules. Like I said, look at your neighbors. If maybe if half your neighbors are layer 1 and half your neighbors are layer 2, then maybe you're more likely to be an intermediate type between those, etc. That's something that can happen even if you don't have, it's not actually learning a bird's eye rule, like where it's saying I'm drawing an outline of the spinal cord and filling in every dot. It's basically just using these repeated patterns to kind of make these corrections. So we definitely want to examine more how this works in tissues that have no clear structure. I think that's part of where I was getting, no conserved structure rather. That's sort of part of what I was getting at the end, which is, if we were to be given a series of tumor samples loaded onto these ST arrays, what if, can we have a pathologist go through and annotate them? Like, is there a clear pattern that even a pathologist can parse out? If so, I believe GridNet would be able to learn it, with enough training data. But the more interesting question, I think, is can we take this gestalt of, say, a couple dozen or more tumor samples where we only have partial or no label information and try and make a common coordinate system out of this? So that's an open question if we can do that. But I think that we can do it as long, if there's a clear signal, even if, if you had 4 tissues that appear in seemingly random combinations, but they have a sort of clear spatial cohesion, like they tend to be together and they tend to abut other tissue types in a predictable way, then, then you can learn that pattern.
Yeah, that would be like really interesting. I'm gonna give now the mic to Braulio, and I think this is gonna be the last question. So Braulio, can you unmute yourself?
Thanks. Hi, Aidan. Great talk.
Thank you.
I'm happy to see this work finished. You probably don't remember, but we met back in 2018.
Yeah.
At the New York Genome Center. So congratulations on finishing this. I had a simple question regarding the memory requirements. Are they dependent on the size of the input data set, or GridNet allocates a specific amount of memory each time it runs?
So the way we've done this is, so like you said, I think if you kind of run it by default, it will try to, there's 2 ways basically to conserve memory in GridNet. There's one by just choosing your, batch size, and honestly, in batch size in the sense of how many arrays it looks at a time. Generally, that will be 1 array at a time, and you can simulate larger batches by doing gradient accumulation. But yeah, the way that it will control how much memory it uses is by picking how fine-grained your checkpointing is. So if you're working on a GPU that only has like 12 gigabytes of memory, you might want to set you might want to set checkpoints that look at smaller chunks that remember sort of smaller chunks of data. So it's something that the user has to kind of play around with. I haven't thought of a way to do it super reliably, but what you would basically do is you consider your input and the size of your input after you've done the subsampling. So, if you're looking at a Visium array that has, 4,000 spots per tissue, and each spot is 128 pixels squared, do the math on that and see how big that is as a whole. And if you think that that's not going to work, say, right, I'm only going to process this in chunks of 50 patches at a time, and to limit how much I keep in memory. So it's kind of I've allowed the user to set the chunk size of the checkpointing to control this, but I don't think there's a formula that I have to say, like, if your input's this big and your GPU is this big, then you should set it at this size. It should be pretty quick to see if if you get a CUDA out of memory error, then lower the chunk size and try that. That's the way I've been doing it, at least. Maybe if more people use it, I can think of a better way.
Okay, great. Thank you.